Right. Yeah. So, okay, Piero Carninci, I'm from RIKEN in Japan, and so I end up there many years ago because I really wanted to develop technologies to analyze the genome. And I went there because my institute, RIKEN, was really starting this new wave of genome project inside RIKEN. So, and I followed the philosophy of developing technologies, particularly focused on capturing full-length transcript, full-length cDNAs, then developing a method called CAGE to analyze initiation site and the profile cells. And using those methods, we have been going through discoveries of genes, protein coding genes, non-coding genes, promoter enhancers, and we had a very long travel in the new world of non-coding RNAs. I'm going to mention all of those aspects in the course of the talk. So I mentioned technologies. One of the key methods is called CAGE. CAGE means cap analysis gene expression, and the CAGE essentially focuses on sequencing at very high throughput this part of the RNA. So what we do, we capture this with a method called CapTrapper. I omit all the details because it was published many years ago. But essentially, we sequence either with Illumina or with the heliscope. We sequence and profile gene expression. So it works in this way. So if you have four RNA samples from hippocampus, somatosensory cortex, or visual cortex, or cerebellum, we see that there are different promoters. So hippocampus, hippocampus specific, for instance, is here, so, and other promoters are more downstream in this gene. So if you ask what is regulating RNA's expression in the gene, the answer is that it depends very much on which tissue we are looking at. And clearly, transcription factor binding site, epigenome, other regulation will all be different for all the different set of samples. Sometimes we will see different protein isoforms that are coming out of those alternative promoters. With this tool and other tools, we decided to, several years ago, to construct a very comprehensive map of transcription and promoter. So, and we started one with the series of the FANTOM project. FANTOM means functional annotation of mammalian. And the idea was to create a very comprehensive collection of promoters from as many as possible primary cells. So we started to collect. My colleague Alistair Forrest has been collecting about 3,000 samples, about 1,500 contains more than hundreds of primary cells and many human tissues, all replicated for the human samples. With the idea that we will use this and create a catalog of promoters, the largest catalog of promoters ever, and the big expression map. And for a subset of them, we also did the time course, a very high density time course, to see at a very high throughput how cells react and if there's anything common in the reaction of those cells. So to summarize, FANTOM is a catalog of expression of many cells. We have a CAGE, so essentially capturing promoters, and we can assess for each promoter transcription factor binding sites that are specific for a given cell type. So we can assess which set of transcription factor is most likely to control many aspects of the phenomena, many aspects of the biology of each single cell. We also have, for a subset of cells, deep RNA sequencing, so we can map a structural transcript, small RNAs as well, and with all of this we can predict also new protein coding, non-coding RNAs, a little bit of splicing isoforms, and so on. So we wanted to make a catalog of promoters, but sometimes when we want to make, you have some idea in your mind what will come out, we have some surprise. One of the surprises is that if you look at the promoters, our textbook image of a promoter is that a promoter has a D starting site. But if you look at the real case, sometimes you see that over a broad area, you can see a lot of initiation sites. All of those purple peaks are different initiation sites. The first group is for 3 different samples from 3 donors from astrocyte. But then you see some more upstream from CD4, CD14, CD4, and finally some encoded cell line here. So the same, what we call the same promoter, uses different set of initiation sites in different cells in different tissues. So each promoter very often have a sub-component or sub-promoters that are active in different cells and are controlled in different way by different epigenome situation or transcription factors. So as a model, you can see that actually you see that there are different modules within the same promoter. If you count here 1, 2, 3, and 3 groups here, we have at least 223,000 of those groups or those initiation sites in the human genome. The mouse is a little bit smaller, not because the mouse is more simple, but because we did not sample as deep in terms of variety of mouse samples. Again, we wanted to make a promoter, but then we had more unexpected funding out of that. In particular, we start to see a lot of enhanced RNA. So if you look at the genome, within the space of a nucleosome, within 150 base pair, you see that there are bidirectional RNAs. One goes left, one goes right. So and there is a space of a nucleosome here. Chromatin marks is H3K4-methyl1 is high, H3K4-methyl3 is low. DNA hypersensitivity sites are here. So essentially, everything suggests that those are enhanced RNAs that are either in introns or far from gene, upstream, downstream in multiple combination. And actually, importantly, in this collection, because we cover a very large variety of primary cells and different tissues, a good part of them are cell-specific and tissue-specific. For instance, if you see those RNA that coming from B cell only, we can see that also DNA hypersensitivity site, H3K27 acetylation, H3K4-methyl1, those epigenome marks corresponding to an answer are also high in B cell. The same is true also for CD4 and natural killer monocyte and all the other cells. So in conclusion, we see that we can also identify an RNA that are tissue-specific. We can even cluster them, and about 60% of 65,000 enhancers are fairly cell tissue specific. This is also quite important if you look in the ENCODE consortium. There is a larger set of enhancers, but many of them are coming from cell lines, and they do not cover the variety of the FANTOM catalog. Human enhancer can also recapitulate the expression in zebrafish, so even if there is no obvious conservation of long stretch of DNA, they can still, in about 50% of cases, recapitulate expression in a specific fashion. Here, in this example, we see expression of a reporter gene in neurons as it is predicted for human tissues. Is this important for all the people in this room? Is this important for genetics? Of course, many of you, of course, are very familiar, been involved in GWAS SNPs, and we all know that they are not mapping. That very frequently they are mapping outside the genes. So those that map in exons are relatively limited. But if you look at extended part of the genes, like promoters identified in the FANTOM consortium, we can see a larger number of GWAS SNP mapping on some functional elements, so we can start to infer possible function. Of course, we are not supposed to read all the names of the genes here, but all of this was published already, so you probably can refer to the original paper. And even in the enhancer, we can find even a larger number of GWAS SNP that are mapping in enhancer. If you look at the co-expression of enhancer and promoters, we can start to infer which enhancer is correlated with which expression of which gene, and perhaps we can start to formulate some functional hypotheses on those genome elements that may be functional, and we have not been analyzing them so far. And it's not a very large number. It's about 65,000 novel RNAs that were analyzed in this study, and they can cover quite a large number of GWAS SNPs. Some example, this is from IBD, and I will omit some of the details here, but I'd like to show you how some screenshot on some example of a promoter that is represented here, that is regulated by many enhancers identified in the FANTOM 5 projects. And in particular, there is in one enhancer here, there is a GWAS SNP that is associated with Crohn's disease. This is a Crohn's disease associated SNPs. And actually, by analyzing RNA data, is possible to identify a link between this enhancer here and a promoter here. All the data can be found in the FANTOM web page. You can Google FANTOM, and you'll probably end up here. And the FANTOM contains various data, including a browser. It's called Zenbu. Zenbu in Japanese means everything. So we have we are trying to put a lot of data, particularly all the libraries and as many additional data as possible. There is a lot of pre-digested analysis, including transcription factor binding site, transcription factors that are specific for cell types, and networks that may be controlling a specific cell type. And also you can find all the raw file. You can download everything, repeat all of your analysis, and we really welcome everyone to really use this data and and include as much as possible in your work. So I mentioned FANTOM, and I didn't say that FANTOM is a project that started many years ago. Actually, FANTOM started in the year 2000, when we really want to annotate for the first time mouse cDNAs. And we went on annotating transcripts until in 2005, in the FANTOM 3, we identify that we identify a lot of unexpected RNAs that were not encoding for any protein. Actually, we found that more than 70% of the genome is transcribed, and more than 50% of the RNAs are non-protein coding. So we call them non-coding RNAs. And perhaps at the end of the talk, we may rename at least some of them as regulatory RNAs, or structural RNAs, or RNA that have some other type of function. So to recapitulate the finding of 2005, was that about 53% of the non-coding of the transcriptome analyzed by cDNAs, at that time full-length cDNAs, was considered to be non-coding. For most of them, we knew nothing, but I put here some regulation arrows, because for a few of them that were characterized over the years, little by little, many groups start to identify some function. They may interact with the chromatin, they may regulate the epigenome, they may regulate translation, they may be structural RNAs as well. They can often do many things. But still, we do not know much for about 95% of them. If you look at the protein coding genes in humans, we know that there are some PubMed entry for the majority of them. But for the majority of non-coding RNAs, there is no paper, no analysis, no work whatsoever. And so we were also trying to address the function of those RNA. So I mentioned about the 2005 data, and just to mention our current estimate that was published last year. And by analyzing the number of genes in the FANTOM 5, where we have all the cage libraries from 100 primary cells, and in total about 3,000 human libraries, by putting together transcripts, not only transcripts, that have a very clear cage peaks, we identify the promoter. And at the same time, we also have some RNA-seq connected to that. And on the promoter, we found also some epigenome marks. So we are quite confident that they are true transcript by any, by any type of analysis. We define a set of robust non-coding RNAs, about 28,000, for which we are quite sure. If you really want to be very stringent is 9,000, and very aggressive is more than 60,000. So this is the FANTOM cage annotated transcript that expands quite a lot the gold standard that is GeneCode. So it's about, in total, is about 20,000 novel non-coding RNAs. And in our opinion, and we are talking with GeneCode actually, there are 7,000, 8,000 of GeneCode genes that need a better annotation at the 5' end or need some refinement. Actually, we are working with GeneCode at the moment to really integrate those analyses. So are those useful? So I put here some, so because for all the non-coding RNAs we know the expression, we can overlay non-coding RNA that are associated to some, are mapping over some GWAS SNP to the expression of the non-coding RNAs. And that's what we found very often, that we found a very nice correlation between, expression of specific tissue and diseases. So we often find that, for instance, brain is responsible for, is the tissue which longer non-coding RNA are expressed and are correlated to dementia or autism and so on. Okay, so there is not only GWAS SNP on non-coding RNAs, but also we can correlate with specific expression in specific tissue where we know that the disease takes place. We'll meet this, and I will just to mention a little bit a summary of the possible function of the non-coding RNAs. We have been looking at EQTL and also at GWAS, and we have about 3,000 and 2,000 each. So those non-coding RNA that may be linked to some human disease. Of course, all of this has to be proven, but I think we have some more tools now to test that. Also, we've been analyzing conservation. So conservation is a tricky thing, and but sometimes we have conservation at the initiation region. So only promoter is conserved. Sometimes both promoter and exon are conserved. Sometimes only exon are conserved. So based on all the conservation or correlation with the GWAS or EQTL, we believe that out of 28,000, perhaps 19,000 of them may have some sort of function. And of course, again, this is only fair, then we need to really do experiment at a certain point. So I would like to mention a group of non-coding RNAs. Antisense very often do have a function. It is a little bit easier to prove the function for antisense RNAs, and I have a few slides about the project that has developed out of studying antisense. And we started in the year we started in around 2008-2009 to look for antisense of UCHL1, that is one of the parking. And we found this antisense of UCHL1 that is a non-coding RNase that is overlapping the initiation site. And so we started to do some experiment, and the most obvious experiment in collaboration with a group of Stefano Agustincis at SIS and now at IIT in Italy. We start to overexpress the antisense in cells that do not express the antisense RNA, and we wanted to see if we can see some effect to the sense level. So we all know that sense-antisense might regulate expression level, and we try to see if there is anything at expression level, but we saw that actually the level of expression of the sense is essentially unchanged. So we were a little bit disappointed, think that this is not functional. But before going to the next step, we decided to see what happens to the protein expression. And knowing all the siRNA, microRNA, we would expect that some antisense will decrease protein expression. However, we were very surprised that by adding the antisense, we can see much more protein. So there is no change at RNA level, but increase, dramatic increase of protein level in the presence of the antisense. So, well, this is unexpected, but we started to map, and we identify, by computational analysis, we identified that in the antisense, in the non-overlapping part, there is some sign B2 element we call inverted because it is inverted with respect to the sequence in the database, represented here in green. And so we start to make deletion mutants. We took away some unrelated ALU element, and we took away, we deleted the sign B2 element, or we inverted. Only when we inverted or deleted the sign B2 element, we saw that our beneficial effect in to induce protein translation disappeared, suggesting that the sign element in the non-overlapping region was mediating somehow enhancement of translation. Also, it's not shown here, but also the antisense region is as well important, and we have many more controls and other studies to validate this. So with Brunella, actually, they've been checking if can we, for instance, use this antisense to induce protein translation in vivo. And actually, they studied the COX-7b that is responsible for microphthalmia with skin lesion, and using the Medaka fish. And the model is this. So they put a morpholino to inhibit splicing and leaving about 30% of wild-type RNAs, and about 70% of RNA bypasses the exon 2. So causing a non-functional protein and actually a very strong phenotype. We designed collaborator of mine designed a sign-up and to having this antisense region and our sign B2 element. And actually what happens is that we wanted to test if this RNA, RNA overlapping the starting site containing a mouse sign element would rescue, increase the translation of a proper protein from this remaining 30% of RNA. And actually, in 50% of the cases, this worked very nicely. So essentially, it is possible now to rescue a deficiency of proper RNA by enhancing protein translation using one antisense RNA. So this is very promising, we think, because we can target many genes, many haploinsufficiency. We in my lab and in collaborators' lab now we are actually trying to use those antisense RNA, changing the antisense region to target genes that originally do not have such antisense, and increasing the amount of protein in case of haploinsufficiency or any other disease which will need some increase of translation of protein. So this is one example with one class, but we, in the continuation of the FANTOM, we have many more long-coding RNA for which we want to assess if there is a function or not. So remember, there are about 20,000 of them that are either conserved or show some correlation with GWAS or other genetics data. So we would like to screen directly if they have a function, and we came up with a strategy, with a principle to perturb long-coding RNAs and see if there is any transcriptional phenotype. So what we do, we put one antisense. Usually it is a gapmer that through RNase H will, for instance, cleave this non-coding RNAs. The idea, if this non-coding RNA, for instance, is involved in repressing a locus, we will see that a gene will be expressed after we knock down the non-coding RNAs. So we look for those responses by essentially knocking down non-coding RNAs, extracting RNA, preparing a cage library, and sequencing and analyzing if anything has changed in the network. So essentially is a transcriptional phenotype what we are analyzing. Sometimes we see very obvious effect in cell growth, so this is a control, but sometimes knocking down non-coding RNA, we see a dramatic effect in cell growth. Sometimes we see even apoptosis, and we see a lot of differential phenotypes just by looking at the cell. But we also wanted to look what happens at transcription level, and we see that very often genes going up or going down. We can see very often. In this example is all using human dermal fibroblasts, but we see that those cells are trying to do many different things. Sometimes these extracellular matrix genes are changing up, down, or up. Regulation of transcription, sometimes p53, cell division, mitotic nuclear division, translation, so on. So we were very much afraid that at the beginning of our project we will see mostly double-strand interferon, stress, and so on, but actually we see quite a bit of diverse response, suggesting that many of the non-coding RNA are actually involved in different aspects. So of course, this is all ongoing studies. A little bit about the networks in the summary. So it involves quite a. I mean, we found non-coding RNA involved in various aspects of regulation of the biology of those cells. So finally, I would like to mention, we look only here on the right, the non-coding RNAs are very often nuclear, and if you by analyzing the partition, by preparing libraries from chromatin, cytoplasm, and other RNAs that are in the nucleus but not attached to the chromatin, we see that many of the non-coding RNAs tend to be in this area very often associated to chromatin. So they may never leave the nuclei, and they may be attached to the chromatin. So we thought that perhaps we should have some direct method to measure interactions of the non-coding RNAs, and we decided to develop another technology to really create a comprehensive map of all the non-coding RNA with all the loci in the cell. So the idea is that we know that many non-coding RNAs do a lot of biology when they are interacting with the genome. So for instance, X inactivation is mediated by X-cyst. Hot air is moving from one chromosome to a different chromosome to modify the epigenome. Many imprinted loci are produce RNA that are bound to their own locus and interact. So we decided we should map them all if possible, and we came out with a strategy. Actually, took from the beginning of the project. Actually, took something like 7 or 8 years of different postdocs. It was really a very tough project to develop all the conditions to go from this initial complex that is genomic DNA, histone, and any other protein and RNAs represented here, to a library where we can map all of them. So we came out with the idea that in the nucleus we will cross-link with 1 or 2% formaldehyde. Then we will cleave the long fragment of DNA with DNase 1 and digest the nascent RNA. Those RNA that have been transcribed actively in any part of the genome. We will digest with RNase H. And then we will put this linker. This is the key part of the technology. This linker is very specific because it ligates RNA with T4 RNA ligase 2 at one side, and the other side ligates is double-stranded DNA with and here it ligates only the genomic DNA. After we obtain this ligated product, we decross-link, digest everything, and prepare from RNA is converted into cDNA, and then after cleaving with the enzyme which cleaves 27 bases into the cDNA, 27 bases into the genomic DNA, everything is ligated to Illumina linkers, is sequenced. We sequence about 1 flow cell of high-sec to 2500 per experiment, and we map RNA chromatin interaction at a very high throughput. So I have many more slides, but I just would like to show some of the samples of the slides. So this is a gene, is a KCNQ1 opposite transfer, which is essentially antisense non-coding RNA, which is imprinted. This is very well known to bind its own locus, not only at its own site, but to distribute along the chromosome. And actually, our methods captures those interactions. In a control where we block RNA polymers for many hours before we do the experiment, we see that those interactions are somehow stable, so they can be detected, even if not as strong, but even after hours and hours, we block RNA pull through. And I'm going to omit this additional controls here with a non-cross-linked sample here. How does it look like? We found that many RNAs, and actually the surprise that many protein coding RNAs as well, many intron of protein coding RNAs as well, they are binding the chromatin, and sometimes is counterintuitive. So they are transcribed from a location here, we, for example, on chromosome 13, and we expand here the target site on the chromosome 13. We see that sometimes they bounded their own locus, but sometimes they also distribute in various regions along the chromosome, and those patterns are different. Some of those are RNAs spread along in cis very, very broadly. Some other are very narrow, and so the distribution is very far from being random, suggesting that there are quite a lot of specific interaction. When those arrows go to a different chromosome, we see also sometimes coding or non-coding RNAs that go to a different chromosome in trans. All the function of this is not known, but the suggestion that the chromatin has much more RNA, and many more intronic RNA are bound to the chromatin. Many more RNA coming from a repeat element are also involved in doing something in interacting with the chromatin. This is some view of a chromosome. It reminds you, it reminds me at least a little bit those high C map where we were in the IC. We see all those DNA interaction map. But here is a DNA RNA interaction map with some of those RNA that spreads along the chromosome very broadly. Some other that show some more specific interaction patterns. Just one more slide about those type of interaction. We find that some of the interaction are, but not all of them, but at least some of them are mediated by possibly mediated by triple helix. For instance, MEG3 is known to bind with by AG stretch, and actually also we found that a similar consensus in the interaction that we have seen with our methods for the MEG3. So perhaps some Hogstin triple helix is at least responsible for the interaction of some of those RNA to the chromatin. Of course, for now we are going to actually prioritizing the knockdown for the non-coding RNAs. We're going to expand those libraries into many human cell types to really understand if those non-coding RNA may be mediator of the epigenome response. So all of this was done at the moment with bulk cells. All the FANTOM is done with bulk cell. Even if we do everything with specific homogeneous primary cell types, we really need to move the field into the single cell both for the analysis of gene expression, protein coding genes, networks, but also for the non-coding RNAs. It's going to be very, very important. And we've been developing technologies to profile single-cell expression by also capturing non-polyadenylated, that very often are the non-coding RNAs. So essentially, we have been putting at first in the fluid C1, where we can profile 90 cells per run. So we have been using random primers because very often non-coding RNAs are not polyadenylated, and enhancer RNA are not strongly polyadenylated, and we want to see them in our analysis. And actually, by making libraries, I will omit all the details here, but the key point is that we have a random primer and it does a template switch here to capture the initiation site. This is a capture actually like a cage, and that will capture promoters, but also will capture the promoters of non-coding RNA and enhancer RNA. In this example, this is an analysis on one enhancer RNAs, and we know that RNA makes a bidirectional transcripts, but we never know if in the same cell RNAs are always in one direction or in the opposite direction. And that's why actually our data seems to suggest that in different cells, enhancer RNA are either transcribed in one direction or in the opposite direction. The cases where we see both expression in both orientation is fairly low, is fairly less. So suggesting that the RNA polymerase tends to land always in the same cells, or at least in the same chromosome, in the same orientation, either one side or the other side. Of course, we didn't prove that with structural study, but it's most likely to be the case. This is 96 cells. We know that 96 is not enough. We move this to the next cage. Again, a cage protocol where we can identify poly A plus, poly A minus, non-coding RNAs that are visible to our methods, but not to oligo-DT priming methods, poly A minus RNA like histone RNA and so on. And with those tools, actually, we are also entering a new era. Is about the human cell atlas. You probably have heard about that. The human cell atlas would like to create a catalog of all the genes, and we will say, from our perspective, non-coding genes as well, all the genes expressed in all the cells of the human body, all the cell types and possibly states, starting from health, but also adding some part of the disease. And the human cell atlas was started in a meeting with Sanger, Broad, Karolinska, and ourselves at RIKEN. Is Sarah Teichmann at Sanger, Sten Linason, Aviv Regev, and ourselves at RIKEN that are coordinating at least a part of this with the broader committee, of course, that is supporting us. And we are coordinating for Asia, so if of course we are in Europe here, but if you are from Asia, we would like to coordinate. Particularly, we have the first meeting with Japan, South Korea, Singapore, India. We would like to include China as well. And yeah, I would like to thank everybody for being here. I would like to mention that no, this is not the end. This is just the beginning. The beginning of a long project to understand the function of the non-coding RNAs and profile their expression in many cells. Again, it's very difficult to thank everybody. This is just a small subset of the key collaborators and some of the funding. And but I would like to really thank all the FANTOM consortium. We put their name, but I cannot really read all of them. And thank you all of you for your attention. Open to any question you may have. Thank you.
Thank you.